Hey, thank you for joining us today. I'm Maggie Kuhl, Vice President of Patient Engagement at the Michael J. Fox Foundation, and I'll be your moderator today. I have done this webinar many years, and it's always one of my favorites. This is our year in review, talking about all the exciting research that's happened in 2024. We have a lot to share with you, so we're glad that you joined us today. Before we dive in, let me cover a couple housekeeping items. If you have a question, you can type it in the middle of the Q&A box in the middle of your screen. Foundation staff and our panelists will try to answer as many as possible during our hour together. To add captions in English, please click the CC button on the bottom right of the media player on your screen. And if you'd like other helpful information, you'll see a resource list with some links that you can click through. If you're experiencing audiovisual quality issues, please check the icon on your screen to make sure you have a really strong Wi-Fi connection to support the live stream. And lastly, if you do have issues or you want to revisit or share our conversation, this will be available on demand at the same link that you've used today, and we'll have captions in a few different languages. So with that said, let's dive in. Today with me are three CMOs. First, we have Dr. Maurizio Ficaris, who is currently Chief Medical Officer at Vanqua Bio, but for a long time was here on staff with us at the Michael J. Fox Foundation as one of our research scientists. So Maurizio, thanks for this reunion on a snowy Chicago morning.
Yeah. Yes, good morning, good afternoon, everybody, wherever you are in the world. Thank you for having me. Very excited to be part of this panel and to talk about exciting things for 2024.
And someone on our panel who has not yet worked for the Michael J. Fox Foundation, but is actually joining us from our offices today because he's there for a meeting, is the Chief Medical Officer at the Institute for Neurodegenerative Disorders and a movement disorder specialist at the University of Pennsylvania, Dr. Tom Tropea. Tom, thanks for joining us.
Of course, thank you, and nice to see everybody, and happy to be here.
And our other CMO, Chief Mission Officer, Todd Scher of the Michael J. Fox Foundation. Todd, thanks for sharing your deep well of knowledge and foundation strategy with our attendees today.
Thanks, Maggie. I look forward to the webinar. I just want to start by thanking Maurizio and Tom for donating their time to share their expertise with the community.
Absolutely. Okay, well, let's jump in and start with some big news this year. The Food and Drug Administration in the United States approved 2 new drugs for Parkinson's disease. These are both to help with motor symptoms and motor fluctuations. The first is Crexont, and the second is Vyalev. And Maurizio, between Fox Foundation and Vanqua, you worked at AbbVie on Vyalev for 9 years. And so you're really familiar with that therapy and this space in general. So maybe you could share with our attendees a little bit more about these therapies and what they can do for patients living with PD today.
Yeah, absolutely. So as you mentioned, those are both medications. So very exciting because again, we are adding onto options and therapies, for people living with Parkinson's disease. And those 2 medications specifically are helping people with the later, more advanced stage of Parkinson's. It's not like, when I think everybody understands that Parkinson's has several phases, and in one where symptoms are becoming a little bit more naughty and resistant to medications. This is where we have motor fluctuations. So both these medications are helping patients to improve those motor fluctuations, reduce those motor fluctuations, improving the time on, so when there's no symptoms, reducing the time off when symptoms are reappearing, for example, so those hours of off time are reduced, or potentially also reducing the time of dyskinesias, which is when symptoms are the medication seems to work over and giving some of those annoying fluctuations, so this dyskinesia. So the good news is that both medications have been approved. One is an oral, is a tablet, is a pill, specifically made to have granules of levodopa, and which we know it's the main drug that helps symptoms of Parkinson's. Those granules are releasing levodopa quickly. Some others are pellets that release levodopa more slowly, so over time. And the good news, the novelty about this medication, this is Crexont, that is coded. It's specifically coded to prevent the degradation of this medication in the stomach and goes directly into the intestine when this medication is absorbed. So is improving the time that this medication stays in circulation and therefore increasing the time of levodopa in blood and dopamine in the brain so they can keep a more stable concentration of dopamine and reducing those fluctuations. Now, as Maggie mentioned, I work on Vyalev, and what is Vyalev instead novel compared to this, like what different compared to Crexont, is that Vyalev is a drug device combination product. So it's an infusion of phospholevodopa and phoscarbidopa that goes under the skin through a small infusion set that is attached to a portable pump. And the difference is that rather than using that by mouth, so still taking pills, etc., it gives a consistent, predictable, continuous infusion of phospholevodopa, which is transformed in the blood directly into levodopa. And again, we are back into the standard approach of controlling symptom for Parkinson's disease. The good news is that is bypassing the stomach completely, right? So any concerns about absorption, or if you have problems swallowing, or in general, or even overnight, like you can maintain that concentration of the drug overnight rather than waking up and taking pills. And this is actually a big advantage. And both studies that demonstrated an improvement in motor fluctuations. I would say that Vyalev has that add-on on the nighttime symptoms, the 24-hour infusion. So overall, though, I think what is important to remind everybody here is that we are just increasing options, right? So giving more opportunities to patients from choosing, what they think is best for them, for their quality of life, for the lifestyle, etc. So very, very exciting thus far.
Great, and that's actually where I was going to go next. So Tom, you treat people with Parkinson's in your clinic. How do you help patients navigate the tools in the toolbox and select which therapy is right for them at the right time?
Well, this is always a conversation with the patient, with their family members, and making decisions around the right treatment is so individualized. Not everyone is best suited for one particular therapy. And I want to highlight something actually that Dr. Fakaris brought up, which is that these are options in our arsenal to expand how we can treat people with Parkinson's and how we can target the symptoms, maybe by reducing the number of pills that you have to take during a single day, or frankly, maybe by not taking pills and instead using an infusion therapy. These are really big advance in the field now to have an option that is a non-oral or non-pill form of levodopa that can be given in a subcutaneous and under the skin infusion, not necessitating sort of a procedure essentially to deliver that medication. So these are really big advances, and all of that contributes to our conversation with patients, right? And so there are some people that will be interested in taking a pill, 2, 3, 4 times a day, and that's great, and we can support people in doing that. There may be people who are interested more in using infusion therapies or other therapies, and so these are just new options that we have. And in my, it's usually early on in meeting with new patients that I will offer education around all these different options that they have. And maybe it's not right at the first visit, but it could become an important consideration at the second visit, the follow-up visits, maybe even years and years later.
Great. And Dr. Todd Scher, many people on the call, everyone on the call, knows that Parkinson's is much more than the motor or movement symptoms that come with the disease. And so we can see on the slide that we have here that there are more than 100 other treatments and testing, many for other symptoms of Parkinson's, such as cognition, depression, swallowing issues. Can you talk to us about where the pipeline is today and any movement we've seen in the last year toward treating other aspects of PD?
Sure. Yeah, I think this is where the optimism on the momentum in the field really comes to play. So as Maurizio and Tom were just talking about, two new drugs have made it all the way across the goal line. Now they'll be available in 2025 for patients, and it's not that common that that would happen. So that's a great achievement. But what we want to see is more options and more opportunities. And you could see that now we have quite a vast development pipeline of new therapies that are making their way all the way through various stages of clinical development and treatment options or the clinical trial options. The big bucket here is on progression. So therapies that are targeting the underlying disease process.
Seems like we lost Todd. Okay, so maybe
directly in Parkinson's.
Oh, great.
And a couple of things I also wanted to highlight is that not all of these are drugs. They mentioned the drug device combination, but we still have innovations happening with deep brain stimulation, which is another type of surgical procedure that's used for people with Parkinson's, and the availability and the vast impact of that therapy is improving based on some of the research that's being done. There's stem cell therapies and cell replacement therapies that are moving through clinical development. Gene therapy is also another genetic-based therapies are all being part of this pipeline. So there's quite a lot of activity, and it's really great to see a lot of the underlying science that we've been learning about over the last decade or so now being converted into potential new therapies for people with Parkinson's.
Great. And so what I'm taking away is there's a lot of different approaches, be it drugs or surgical interventions or even exercise interventions for a lot of different aspects of Parkinson's that also address the different journey with PD, the different time points that one may be walking post their diagnosis. And I'll also maybe give a plug here that all of these treatments cannot move forward in testing and toward approval and use in clinical care without research participants. So if you are interested in joining trials, then you can visit our Fox Trial Finder tool, which will help match you to the hundreds of recruiting studies looking for volunteers to help test their hypotheses. I want to move on now, and Todd, you pointed out how much of this pie chart is around progression and targeting the underlying biology. Our hope would be that there would be therapies that would stop or ease many aspects of the disease by really addressing what is the sort of root cause of the many symptoms of Parkinson's. And so we're going to talk about targets and give a little bit of a 101 to help move us into the next part of our conversation, because just like the therapies that Maurizio, you shared with us work on the dopamine system, we have many other targets that drugs are addressing to try and stop PD. So maybe you can just give us more of a primer on what is a target when we talk about that.
Sure. So I'll try my best, right? So because there are several, as you can see here on the slide, right, several targets. So potentially a gene or the byproduct of a gene, which is a protein, or a pathway, like a series of protein and enzymes involved in a process in our cells in the brain or somewhere else in the body, right? So we know, I would say that maybe we can recollect as a common denominator to one protein that we are very interested about, which is alpha-synuclein, right? So we all know that alpha-synuclein accumulates in this protein clumps in the brain, and we don't know if it does it as a way to prevent or this accumulation, or as a cause. We really started learning about this accumulation in the brain, in the cells, in the dopaminergic cells, in the cells that produce dopamine. And we heard before how dopamine is important, right? So, but now alpha-synuclein clumps in this dopaminergic cell, and eventually the cells die. So when we say what is the target, why are we interested in these targets, is that we know that there are several ways why this potential alpha-synuclein clumps, right? So it could be because the organelles in the cells are not doing the job of cleaning this rubbish, right? So the trash alpha-synuclein that is free and soluble in the cells, there is these organelles that can clean up, but they don't do the job very well. Or maybe there is another protein or a gene, that is involved in producing too much of this protein. And so we can target the gene by saying, okay, well, shut down a little bit, try to avoid this overproduction of this toxic protein. So there are several, processes that can be put in place to address the baseline problem, right? So, and all of those processes that are focusing on one of those underlying problems are targets. So why this is important is because if you understand what a target is, or you actually figure out what a target is, not necessarily what it does, but what it is, you can develop potential medications that go and interact on that target, right? So, and interact by, as I said before, maybe preventing too much formation of a protein, or cutting that protein up and helping the cleaning up, right? So, or activating the enzyme that helps cleaning the protein. So I'm like just making an example, but once you identify what target is, you can act on it by either mechanism, as we said, pharmaceuticals, so creating molecules that interact with those targets, or by using devices, as Todd mentioned, DBS uses electricity to modulate some of those neurons, and that's a way. It's another way to target, or you can use very even non-therapeutic or non device approaches that are stimulating or producing other protein in the brain or other chemicals in the brain that help with maintaining the balance of the brain. And I'm talking about, we always say that at the Fox Foundation, exercise. Exercise is something that we always know it seems not a therapeutic, but it does work. It does work by increasing level of endorphins and something like that. And maybe those molecules are interacting with those targets and making some benefits physically into our brain. So this is like a big answer, and I know maybe sometimes create more confusion, but the take-home message is that when you identify a target, you can find something that can work on that target, modulate that target, and eventually help with the pathology of the disease, in this case, Parkinson's.
Yes, and it differs. Sometimes you have to increase the target activity, sometimes you have to decrease, sometimes you have to cancel out, sometimes you have to replace. And so a lot of our work goes to study exactly how we would modulate. Todd, I want to go to you next, because Parkinson's is such a complex disease. We've uncovered so many targets, but a lot of our understanding of Parkinson's comes from studies from individuals from European descent. And so we, over the last couple of years, and again in 2024, have really added to this bank of knowledge around the disease biology from populations who have been underrepresented in research. And so what is the value of that, and what is the foundation doing to try and fill those gaps?
I think, did you say Todd or Tom? I'm sorry for that answer.
Yes, I did say you, so please go ahead.
Okay.
Dr. Scher.
So I think that one of the keys we're learning about Parkinson's is that there's not going to be one target or one biology that is at play in every individual with the disease, or even at every stage of the journey of the disease. So some of this biology could be at play at risk of developing the very early stages of the disease, and different biology may come at play, as the disease is progressing. So there's going to be a need to really understand which biology is taking place in which subpopulation of individuals with the disease, and also at what stage of disease. And to really understand this and learn this, we need to study a vast population of people with Parkinson's that have diverse backgrounds, both different genetic backgrounds, different environmental exposure backgrounds, to really understand and fine tune what could be the causes of Parkinson's, and what biology may be at play in the different populations. So, as Maggie was saying, historically, there's been a narrow slice of the population of people with Parkinson's that has been studied mostly from North America and Western Europe. And the foundation took on a big effort with our partners from the ASAP initiative and NIH to launch a more worldwide study that's called the Global Parkinson's Genetics Program, GP2. And they're now in that study integrating and working with patients and clinicians throughout the entire world, Africa and Asia and Latin America, as well as North America, South America, and Europe. And what we're really uncovering is that there's some very clear commonalities in the cause of Parkinson's and the underlying biology in Parkinson's across these populations. But there are some more population-specific genetic findings and other biology. And it's really important, again, to do this in order to develop therapies that will treat the biggest population, the vast majority of people with Parkinson's, to know what really is the biology we need to target and which are the right patients that would benefit from those types of therapies. So we can design the best trials with the best chance of success.
Absolutely. And some of the targets that we've already identified may have broader application, and we may uncover some new ones. So Tom, Dr. Tropea, we see on the slide a couple of the targets, the pathways, proteins that we have identified and profiled to play major roles in Parkinson's disease. Maybe you could just quickly cover some of those.
Sure. So, I'd like to sort of tie together what Dr. Scheer and Dr. Fakaris basically mentioned, which is essentially understanding more about the biology helps us to understand what are targets for drug development. We've done a lot of this over the years, and recently, and I think people on this call have probably seen that there are drugs in development, drugs in clinical trials that target specific pathways. Right. These are, rooted in, years and years of biology and understanding, targets for potential therapies. And so, clinical trials, ongoing right now and in the past couple of years have targeted individuals carrying a variant or a change in a specific gene, and in particular, the LRRK2 gene or the GBA gene. But it's also important to understand that while we use targets as a way to approach, say, enrollment in a trial, it does not necessarily mean that those drugs may not be relevant outside of individuals carrying changes in those specific genes. So again, we understand the target so we can develop therapies. Understanding more about the biology helps us to understand if targeting that therapy is relevant even outside of, say, a group carrying a change in one of those genes, right? So it's important to understand that. And we have, some really interesting, studies ongoing, clinical trials as well of drugs in development, targeting, gene pathways, targeting alpha-synuclein, the protein, most associated with Parkinson's disease symptoms, as well as inflammation. And so I think in the next bunch of years, we're going to see a lot of interesting results from those studies. It'll help us to understand, again, more about the biology and how those targets relate to individuals versus Parkinson's disease much more broadly.
Maggie, just one thing I also wanted to add on this. One of the things that's very important and exciting about targeting this underlying biology is that these are the processes that we believe are involved in the onset and progression of Parkinson's. So the hope here is that by interfering in these key biological pathways, you may actually be able to interfere in the actual disease process and progression itself. So it's different than some of the therapies that we've talked about that just came across the FDA goal line that are managing and mitigating some of the symptoms of the disease. With all the advances in understanding the underlying biological pathways and targets of the disease, there's the hope in the design of these trials to really be trying to target the disease itself, the actual nuggets of the disease that are leading to the symptom development and progression.
Absolutely. And Maurizio, anything to add? I know that you're working in this space particularly.
Yeah, so two things that I want to do. One is to close the circle, to what Todd mentioned. Even the symptomatic therapies, that is all we have right now for Parkinson's disease, started by focusing on a target, right? So we initially realized, long ago, that what was happening in Parkinson's was this reduction of dopamine. And here we are, right? So we couldn't give dopamine directly because it doesn't get into the brain, but we were able to give levodopa. And that was the start of therapy levodopa-based therapies that we know are, the cornerstone of symptomatic therapy from 1967 on, right? So more than 50 years now of levodopa available therapies. But that started with a target. And speaking of those targets that are instead now of interest because they can really slow down the progress, or the hypothesis, the goal is to slow down the progression of disease or even potentially stop the progression of disease, making this, the disorder from progressive to chronic, just keep, as it is, or maybe preventing it in the future, is focusing on those mechanisms that seems to be the underlying reason for the degeneration of those neurons. And I give an example, and I want to close, returning to what Dr. Tropea said before, which is there are targets like the GBA, and as you said, I'm interested in that, right? So it's only 10%, approximately 10% of the population with Parkinson's disease had a mutation on the GBA, and that GBA gene encodes for a protein that is in the lysosome, which is one of the organelles that cleans up, like as I said before, the toxic proteins. But what we learned is that everybody in Parkinson's, the majority of patients with Parkinson's disease, has a reduction of that protein that cleans up the toxin proteins, right? So that cleans up alpha-synuclein. And so you don't have to have a mutation to potentially have that reduction in GCase, that's the enzyme that encodes, by GBA. So again, the importance of understanding the targets or finding targets is because you can learn the biology underneath, the progression of the disease, and you can target those targets, potentially via pharmacology. So for us, absolutely very interesting because it's open opportunities for truly slow down or stopping the progression of disease, which is, the goal that we all have.
Yeah, I like to think of it as sort of like a GPS or a map, right? If you can put in a starting origin and a end destination of Parkinson's, it might show you the roads to get there, but other people might get on that same road just from different entry points, different on-ramps. And so, as you said, if you treat the disease pathology that's on that road, if you came from one spot with a GBA gene variant or another spot with exposure to Agent Orange or other things that we know send you toward Parkinson's, if you're treating something a little bit more downstream, it might have efficacy in different causal populations.
Right. And as Todd, said before, it might not necessarily be a single approach, right? So we obviously, for developers, you need to focus on one thing at a time. But I believe that in the future will be a combination of all those strategies, right? So that's why we're not going to put all, the eggs in a single basket. We are truly focusing on multiple approaches because potentially it's a combination of all of that, targeting all of them, that will bring us to the conclusion.
Yes, yeah. And even though there's a couple on the screen, we have more than 100 targets that have already been named in Parkinson's disease. We are not stopping the pursuit of new targets, or we're working very hard to sort of nominate which targets we think play a really big role and put our resources toward moving those closer to drug development. So now it's time I'm going to make a small call out because research like this that we're talking about at the foundation is made possible by our community. Giving Tuesday is coming up. So we encourage you to think about giving to the foundation, and all gifts made before or on December 3rd will be matched up to $4 million. So you can double your impact. And any gift is a double investment in our biggest, boldest initiatives to close in on a cure. So there's a take action box on your screen that I'll direct you to. And while I'm doing my commercials, I'll go back to my previous one as well, because there are, as we've talked about, there's so many targets, there's so many therapies. A lot of trials were in development. They recruited volunteers, they followed volunteers, we're now getting the readouts. And that means that a lot of them are going to be starting and moving toward this next phase, which is really exciting. It means that they need a lot of volunteers. I know my team, which works on recruitment, has a lot of calls with companies who are throwing out numbers like, we just need 400 people with Parkinson's. And so there's multiple studies coming down the line. And so if you are interested in helping us move closer to a cure in that way, again, Fox Trial Finder is a great way, or you can keep an eye on the Fox Foundation communications, because we'll be sure to let you know when some of those big trials start opening. So want to move us forward in our conversation to how we can get the right drugs to the right people at the right times, as we have been talking about, and really as quickly as possible assess, are these the right therapies that are doing the things that we want to on the targets and having the right impacts on disease and how you all really feel day to day. And so for that, we have had some breakthroughs in biomarkers over the last year or so. And so, Tom, I'm going to start with you. And again, sort of go back to our one-on-one. What is a biomarker? Why are they so important?
Sure. So biomarkers can take many forms. So a biomarker itself is something that's measurable from some fluid or process throughout the body that we can access and measure. So that's really broad, right? Like that's not actually all that helpful, but you can think of it as maybe like a protein that we can measure, say, from the blood or the spinal fluid. Or a biomarker in a really broad sense could be, say, even maybe an imaging marker. That's a little bit expanding what a biomarker traditionally is, but it could be an imaging marker of a process that's that's going on in someone's body. It's basically an external measure of an internal process, right? So you can think of that. It's really helpful for us to have effective, easily measured biomarkers, because it allows us to understand more, again, about the biology of what's going on in someone's body. But it can also be an indicator of an ongoing process, right? So we may be able to measure these, say, proteins or whatever we're going to measure over time and see how things change or may be affected by an intervention or a drug or a therapy that we use. Maggie has alluded to some really big breakthroughs in biomarkers this year. I don't know if you want to talk about that more. But
yeah, please go ahead. You're really involved in our Parkinson's Progression Markers Initiative, which has been behind some of these. Oh, I always forget which, it's like a mirror behind me there, our PPMI sign. So yeah, tell us about what's happened.
Absolutely. So, this year, I hope everyone has seen some of the, excitement around our ability to measure and detect the pathological protein alpha-synuclein from samples provided from spinal fluid, as well as from other tissues around the body. This is really a breakthrough. And in the past, we have relied predominantly on a diagnosis that is justified by symptoms, clinical symptoms and signs that we see in the office. And we've moved from our ability to do an exam and give a diagnosis, maybe with some imaging, to detect the biology of Parkinson's disease, right? So rather than clinical symptomology, we've moved to our ability, we've advanced in our ability to detect biology. Has so many implications for what we can do from studying the biology, studying, drugs in development. But also, I think it has a big impact on how people can define their own disease and their own symptoms, right? It can be helpful to know with more clarity why you have, say, tremor or changes in your walking speed and things like that, that are beyond just, the clinical diagnosis that we've relied on for so many years. So there's a lot of impact on this big breakthrough.
And I'm gonna just define quickly, because we've been using the term biology of PD a lot, and we got in the Q&A, what does that mean? And I think your definition of biomarker, which is, I'm going to apply perhaps even more broadly to disease, an internal process. So like you said, there's the clinical experience, there's that you have tremor or that you have depression and or your memory and thinking issues, but then there's actually what is happening inside that is causing those manifestations. And so that is what we mean by biology of disease when we're talking about what targets are at play or what changes you can measure, like we're talking about now. So wanted to level set for that. But Todd, as Tom was saying, we had this biomarker breakthrough with our PPMI study, and now we can detect this disease biology in living people with the disease. And then this year, we had this letter from the Food and Drug Administration that really endorsed this biomarker. Can you tell us about that milestone and why that is important?
Yeah, I just want to add a little bit about the biomarker. So first, for the FDA support, this is really the FDA encouraging the use of this biomarker and further studies utilizing the biomarker to help better inform how clinical trials can be done in Parkinson's in the future. And I think that's a key step because we want to have trials designed that have the best chance for success. So having the best tools so that we can, again, design the trials so that if we have a therapy that works, we actually can detect that it's working. One of the most important things also about the biomarker is that right now this alpha-synuclein test is just a yes/no test. So it just tells you whether you have the presence of the synuclein or not. It doesn't tell you how much is there. And it also relies on a spinal tap, or it's measured in CSF, cerebral spinal fluid. So the foundation, it also has quite a robust effort now to improve this biomarker, both to make it quantitative, which means rather than yes, no, it actually gives you a number readout so that we could see, for example, does it change over time in an individual? And importantly, if a therapy comes in, does it reduce the presence of the synuclein, which would indicate to us that it's positively impacting the biology of the disease? And also to see if it could be moved to a more easily obtainable biological samples, particularly with a lot of effort to see if we could develop the measure in blood, which could be then done at a much broader scale in the population. So this is, an incredibly important advance because it allows us right now to better design clinical trials using the biomarker, and also opens the door for much more robust study of the disease and improvements in the biomarkers going forward.
Great. And Maurizio, maybe you can build on that from the industry angle. How do drug developers and study sponsors, trial sponsors, think about biomarkers and their use?
I think Todd set the ground very well, right? So I give examples like how do you diagnose diabetes, right? So we know that diabetes is a chronic disorder that can cause clogging in the arteries, can give neuropathy. You can have several disorders, but it's easier to detect because you do a blood test and you have glucose, sugar in blood, and if it's a higher, too low, you can actually adjust that, and you create therapies based on that marker moving in the blood, so you can control, if your therapy is working well or not, right? So, and as Tom mentioned earlier, so far, even with the medications that we have available, you can say it was working or not working because it's based on symptoms. So it's something very easily to assess, right? So you're shaking or you're rigid, you take levodopa or you take another medication that is working for symptoms, and the symptoms disappear for that period of time, or now for a longer period of time because we learn about new medications. But point is, you can immediately know if this medication is working. Now, when we're moving towards disease-modifying therapies, so something that can really is focusing on slowing the progression, well, unfortunately, if we don't have something to measure, as Tom mentioned, like biomarker is something that we can measure, whether it is in blood, in the spinal fluid, an imaging marker, a digital marker, something that you can, move, like something that you can measure physically. You, as a drug developer, you only rely on clinical symptoms, right? So, and if we are working towards slowing the progression of disease, we are going very early in Parkinson's, just after diagnosis or potentially even before diagnosis, right? And if we need to rely only on symptoms, the risk is that it's going to take 5, 6, 7 years to notice a difference. And that is, one, very costly. And I don't think patients are actually really willing to wait that long, right? So that's the sense of urgency that we all share. We as drug developer, physician, because they see patients, the foundation has been a mission of this, right? So we have a sense of urgency. So finding a biomarker is a way for us to detect, as Todd mentioned, if the medication that we're testing on that biological background, in the biological basis, like is working on clean the alpha-synuclein that we heard, or other potential mechanisms, is that working by looking at those measurable things in blood, spinal fluid, imaging, etc. And if we can, like we need a lot of data, unfortunately. That's another thing. Like before we can use a marker to define whether something is working, we need a lot of data to make that marker qualifiable, to say, okay, yes, if we see a reduction of alpha-synuclein, then is actually targeting the biology underneath Parkinson's, and potentially that medication works. And there are several markers, right? So we heard there are several biomarkers. You can use biomarkers to enrich the population, meaning like diagnostic marker. Then you say, oh, then that patient truly has Parkinson's and it's not multiple system atrophy. It's not another disorder. Maybe that's like, so you help narrowing down the population. And there are markers for progression, which is the most important, well, it's not the most important, but a very important thing for drug development. Because if we need to rely only on clinical, again, as I said, it will take very long and it will be very expensive. It will take a lot of people, not only 400 people, as Maggie mentioned before. It will take thousands of people, right? So, but if we can have a reliable marker, then we can reduce the sample size and actually come up with the results sooner, right? And with more precision. So this is why it's very important for drug developer to have a reliable biomarker.
So much faster, higher likelihood of success. And Parkinson's is such a complex disease. We've talked about so many things that go wrong biologically, internal processes. Obviously, again, people experience so many different symptoms and rates of progression. And it means that there's a lot of room for opportunity, places that we could stop the disease. But it also means that we need a lot of people to help us learn about all these different subtypes and experiences. So again, like Maurizio, you said, we need a lot of data. We need a lot of people to participate in studies to help us learn, because you are the experts. I think actually Michael was just, in the news doing some interviews and such, and he was saying, if there's a room full of scientists and there's one person with Parkinson's disease, they are the expert on the disease. And so we need you experts to help us understand all of the different intricacies and how we can stop Parkinson's for every person living with PD and at risk for PD. So I really like to save a lot of room for questions on this webinar each year. I'm going to move us forward just one more slide, because we could not talk about news in 2024 without talking about the passage of the National Plan to End Parkinson's Act. And so, Todd, if you could just give us a brief overview of what this is, what this means for people living with Parkinson's and research, and where we are currently in our next steps.
Yeah, I think first, most importantly, the goal of this plan is to keep in mind and remember that the United States government plays a critical role in both the support of research, but also the care of people with Parkinson's disease. And we can't do this alone in terms of moving forward the development of new treatments for Parkinson's. We really want to get more strategic and focused attention from the federal government in the United States. So that was the goal of this legislation, the National Plan to End Parkinson's Act, which was passed by Congress and then signed into law this summer. And it focuses the attention of the US government to develop a national Parkinson's project, something they'd done some years ago for Alzheimer's disease. The result of the Alzheimer's disease Act and subsequent plan led to not only an increased amount of funding and attention at the federal government for Alzheimer's disease, it developed a more coordinated plan across the research enterprise up through drug approvals and into the clinical care and delivery of care to people with Alzheimer's. So where we are right now for the Parkinson's plan, as I mentioned, it was passed this summer. And now, as the next step, the U.S. Department of Health and Human Services will be charged over the next period of time to develop a holistic, coordinated plan for the National Parkinson's Project, which is an initiative to both prevent, treat, and cure Parkinson's disease. So it's a great opportunity for us to get more involvement and more focus from the federal government to join this fight in a more holistic way to improve the lives of people with Parkinson's disease.
We're asking a lot of the attendees on this call, but we have a lot to do and we go much farther together. So as Todd said, there's a lot to be done, and you can be updated on what's happening with the national plan and help us advocate for the support and the policies that it hopes to enact by visiting our website as well. So with that, let's turn to some of the questions from the audience. The first that I wanted to talk to is actually go back to our first topic and these new therapies. Maurizio, I'll toss this to you. Some people may be aware that with long-term levodopa use or advanced disease, people experience dyskinesia, uncontrolled movements. Do these new therapies help address that side effect at all?
Short answer, yes. When it comes to both medications are aiming to reduce motor complications, right? So when I say complications, is a fluctuation, so changes in on and off, unpredictable, as well as dyskinesia. The reason for this is because dyskinesia is not necessarily a problem of long use of levodopa, but it's just when after degeneration of the neuron, sensitivity of the receptor, etc., when you take a lot of levodopa, that causes those potentially unwanted movements. By keeping the concentration of levodopa stable in blood and therefore try to keep the concentration of dopamine stable in the brain, you can prevent those peaks that cause dyskinesia and those drops that cause the off symptoms. So all in all is a way to keep that symptom control as stable as possible during the day and potentially during the night in the case, for example, of Vyalev, right? So this is something that, is addressing, the dyskinesias. Can we say that will prevent from, developing dyskinesia in the future? We don't know, right? So we haven't done any study that shows that people who start with Parkinson's and maybe start taking those medications will not develop dyskinesia. That's something that we haven't studied. We don't have an idea yet. But again, those medications are indicated for people who have a little bit more advanced stages of Parkinson's. So we haven't addressed that.
Well, I guess we will see. It just came to market, and so people will be taking it over the next couple of years. And again, the true experts in the experience. Todd, I'm going to turn to you for our next question, which is what new research has happened this year in our understanding of the gastrointestinal tract's connection to Parkinson's? Perhaps also I'll throw microbiome in there if that's not the same thing in your mind.
Yeah, I mean, this has been a real area of interest, not just in Parkinson's, but in neurodegenerative and neurological diseases broadly, which is the interaction between the digestive system and the brain, what they call the gut-brain axis. So in Parkinson's, it's particularly of interest because there's some data that suggests that early symptoms of Parkinson's may actually begin in the digestive system. And we talked about some of the advances with alpha-synuclein and the importance of alpha-synuclein in Parkinson's. And it is actually found that there are alpha-synuclein pathology in the nervous system that controls the digestive system. So it's not only found in the brain. There's been a lot of effort and interest around what you're calling the microbiome. Microbiome is, we are both an animal itself and we are an environment. So there are bacteria that live within our body and in different parts of our body, and one main area is within the digestive system. So the microbiome is actually the bacterial makeup of your digestive system, and each individual has a different microbiome based on their diet and their genetics and other factors. And there have been some studies that have indicated that the particular makeup of the microbiome may be different in people with Parkinson's disease than those without Parkinson's, suggesting perhaps that there's some involvement of that microbiome in the disease. I think it is interesting because we talk, for most cases of Parkinson's, we say that the cause of Parkinson's is the interaction of your genetics and your environment. And if you think in your body of some of the places where this happens, are both areas that there are early symptoms of the disease. So one is in the olfactory system. So in your smell, which is where kind of some of the environment is getting exposure to your body. And the other is what we're talking about now, which is in the digestive system, which is also a nexus of where environmental factors could interact with your host you're the host, you're the genetics of the host, which is you. So I think this is an area of interest. There's a lot of research to still determine, exactly the role in the cause of Parkinson's, but certainly digestive symptoms are important features of Parkinson's, and a lot of work is being done to also try to figure out how to mitigate those symptoms as well.
Yes, and like Maurizio gave a plug around exercise, I think diet is one where we know it does good things, and there doesn't necessarily need to be a lot of research on exactly, which diet is best. We have a great guide on our website from our on-staff movement disorder specialist, so I encourage you to check that out. But a healthy diet, Mediterranean, fruits, vegetables, healthy grains, healthy fats, et cetera, based. There's some nice outlines in there to help manage life with PD. And Todd, you sort of touched on where I was going to go next, but I'm going to turn it over to Tom, Dr. Trapea. We got a couple questions on factors that could help us identify Parkinson's very early on. And so Todd had talked a little bit about smell loss, and the PPMI study that we support, that again, you're one of the leaders of, is really leading the charge in understanding the relationship to smell loss early in disease. Can you tell us perhaps what we're learning and maybe what some of our attendees can do to help us learn more?
Sure. Smell loss is, of course, I think one of the biggest expanding, area for us to collect samples from individuals and data from individuals prior to the onset of, say, tremor or what we traditionally call Parkinson's symptoms. But even years before that, people who later on go on to develop tremor and Parkinson's symptoms. Constipation is very, very common. In fact, anxiety and depression are very, very common, but these are so non-specific, right? And they can happen to a lot of people who don't end up going on to develop a neurodegenerative Parkinson's, Alzheimer's disease, or something else. And so one of the best study to this point is actually the link between the loss of sense of smell or decrease in sense of smell and the later development of Parkinson's disease. Now, of course, it's not in everybody that develops Parkinson's symptoms that has a loss of sense of smell, but there is a really, there's a strong relationship between the loss of sense of smell and the development of what we keep calling the pathological protein or alpha-synuclein. And so it's now becoming one of the best studied areas. And in the PPMI study and in efforts sort of around the PPMI study, we are expanding how people can go about getting smell testing, right? And so there are efforts led by the Fox Foundation to expand access to smell tests. And this has been, both very helpful for us to understand more about what it means to have loss of sense of smell, but also for reaching back out and saying, hey, we found this, finding in you, would you be interested in coming in and doing more work with us? And that more work may look like participating in a study, or it may be, completing some questionnaires, right? So it serves a whole bunch of purposes for us. It's expanding what we know about the disease, but it also helps to address what has come up a number of times, which is that we have this need to, for participants in studies at all different levels.
Absolutely. And so on that theme of maybe early risk detection or understanding some of the underlying factors that led to disease, we got a question about genetic testing. We've talked a lot about genetic targets. How can individuals, either with Parkinson's or concerned family members, get genetically tested for some of these genes that we have identified to be linked to Parkinson's? Tom, I'll see if you usually counsel your patients in any way, but of course others jump in as well.
Yeah, of course. I'm happy to take this one. And I've spoken in a number of different forums around access to genetic testing. There is no one way that we can offer genetic studies and frankly, genetic counseling to everybody, not only with Parkinson's disease, but actually more broadly and, across all different fields of medicine. There's although genetic counselors do tend to provide a large amount of counseling prior to doing testing, there really is kind of this mismatch between the need and the supply for neurogenetic services. And so there is no one way to go about getting access to testing. There are large centralized programs for people that have a diagnosis of Parkinson's disease, and I would encourage you to seek out those opportunities. They do provide counseling and access to testing. But there are also programs that exist where you can initiate your own testing. You can discuss it with your physician, your neurologist, your primary care doctor about how to access genetic services. And that may be through a local genetic counselor, a neurogeneticist, or a medical geneticist, someone with training in how to conduct neurogenetic services. Or it may be through one of these centralized processes, right? Sign on and, participate in a research study or, expanded access program from one of the companies that provides genetic testing. There are opportunities to do this in clinical, in the clinical realm, meaning just for your medical care. There are opportunities to do this in research. All right. So there really is no one way to do this. And I would encourage you to talk with your primary care doctor, your neurologist, and talk about different ways that you can do that if you are interested in pursuing testing or counseling. One of the most important things is, prior to pursuing testing, is actually talking with someone that is informed, that can explain what it is that you might learn about testing, what you won't learn about testing, and how, if they're interested, what your family members may be able to access as well. So I hope that's helpful. It's actually just it's a really sort of amorphous, it's a very complicated field right now, but there's a lot of effort going into trying, increasing access to testing for anyone who's interested.
Great. Well, we're coming up on our hour. We got so many questions on so many different topics, everything from artificial intelligence to non-motor symptoms to what phase 2 and 3 trials are coming. We wish we could cover everything in so much more detail. Of course, in this webinar series, in our podcasts, on our blog, we tried to go much deeper on a lot of those topics. So if we weren't able to answer your question or you didn't get all that you were looking for today, please visit our website, and hopefully you can get more or tell us where you're still lacking, and we'll put it on our agenda for next year. But I want to end with a little bit of around the room. This is a look back at 2024. I want to now look ahead to 2025. And what are you most looking forward to, or hoping, or see in your crystal ball that this is going to be the year? What should people be looking for, waiting for, partnering on to make a reality? Todd, why don't I start with you?
Yeah, there's a couple of things I'm looking forward to. I mean, one, I think we're going to have more advances with these biomarker tests, which I think are going to really then accelerate our therapeutic development and trials. There's also some very important trials around alpha-synuclein and other targets that we'll be getting the results from this upcoming year. So I think we have a chance for another very exciting year next year. With the momentum continuing to build forward for Parkinson's.
Maurizio, what does 2025 hold?
Well, so I'll second the biomarker, sentiment that Todd just expressed. I'm really hopeful, both because of the clinical trials, as we mentioned, so that the help that can provide to clinical trials, but also optimistic that we will find a way to improve that quantitative measure of alpha-synuclein both in the spinal fluid as well in a different matrix, potentially, something easier to collect than through a lumbar puncture spinal tap. So that I am very, very optimistic, very, very positive. And the second is, I think among the targets that we mentioned, truly, I do believe that GBA, and I know I'm biased, but GBA has been one of the highest interest in the last few years. There have been several clinical studies ongoing. Some of those did not provide the results that they hoped for, but they really shed a lot of light on understanding where we need to target. Again, not only what to target, but where, how, right? So it's not by moving this molecule, is not by moving this enzyme, is actually activating the enzyme directly when it matters. So working towards that, I think is going to be very interesting in the next couple of years. but specifically next year for us to understand how we can improve the activity of that enzyme that cleans up alpha-synuclein in the cells. So this is my spin for now.
Great. And Tom, the last word's yours.
Oh, wow, I get the last. That's great. I'll be very quick. I think in the next year, we are going to continue to refine what we believe to be the biology of Parkinson's by expanding our biomarkers, but also improving those biomarkers and using those in a foundational way rather than an alternate, sort of diagnosis. So that's what I'm most excited about this coming year.
Well, thank the three of you for joining us, and thank you who are tuning in for giving us your time today. We hope you found it helpful and informative, and we will be sending a link to this presentation to watch again or share as you like. Thank you one more time, and have a great day.
Thank you.
Thank you.
Happy